So biomolecules have to analyze chemical composition, then primary and secondary metabolites, then biomacromolecules are proteins, polynucleic polysaccharides, nucleic acid structure of proteins, then enzymes. So how to analyze chemical composition? So comparison of elements present in non-living and living matter. So element percentage, weight of earth crust and human body. So hydrogen, carbon, oxygen, nitrogen and sulfur present more in human body than sodium, calcium, magnesium, silicon present more in earth crust, and silicon negligible in human body, and nitrogen is very little in earth crust. Now list of representative inorganic constituents of living tissues. So sodium, potassium, calcium, magnesium, water compounds, all these components present in living tissue. Compounds are NaCl, calcium carbonate, phosphate and sulfate. Now there is bitter ionic form. Now this one, NH3 positive, CHRCOO negative, it is neutral, it is bitter ionic form. And amino acids, these are alpha amino acids. Why? Because there is amino group and carboxylic acid group, then variable R group, then hydrogen. Now on this side COO negative, on this side NH3 positive. So this was the zwitter ionic form. Now diagrammatic representation of small molecular weight organic compounds in living tissues. So small molecular weight organic compounds. So 6H12O6, glucose, ribose, C5, H10O5. Okay, so ribose. So OH on third side it is upward, and OH COO NH2 and CH2OH. Now on this side OH all downward, CH2OH. So this is ribose. Now glycine, alanine and serine. So hydrogen in glycine, CH3 methyl and hydroxymethyl, right? Hydroxymethyl, that is serine. Okay, so these are the amino acids. These are alpha amino acids. Now fatty acid, that is palmitic acid, glycerol, CH2 OH, trihydroxypropane. Now triglyceride, R1 R2 and R3 are fatty acids. You see NH2, now CH2 CH and CH2. Then fatty acid, you see this one is the fatty acid, that is palmitic acid, contains CH3 CH2 14 carbon COOH. Now the same fatty acid in here like R1 R2 and R3, these are carboxylic aliphatic chain. So these are the fatty acids. Now phospholipid lecithin, component of membrane, cell membrane. You see CH2 CH2, R2COO and R1COO, then CH2. You see phosphate is present, P double bond OOH, then CH2, then N, CH3. N group is also present. So this is the structure of phospholipid, that is lecithin. Now cholesterol having the OH 1 2 3 4 cyclic ring. So this is the structure of cholesterol. It is type of lipid, kind of steroid. So this is it. Now fats and oils, and all these are lipids. Fatty acid, glycerol, triglyceride, phospholipid, cholesterol. So all these are fats and oils, that is lipids. Now there are nitrogen bases, that is adenine, purine, containing 2 heterocyclic ring. Then adenosine, it is nucleosides. You see adenine is attached to the pentose sugar. Then adenylic acid, you see, then the adenylic acid, it is nucleotide. Then uracil, it is pyrimidine. Nitrogen bases is nucleosides, that is uridine and nucleotide. So this is it, you see. Now another is primary and secondary metabolites. So secondary, you see some secondary metabolites. So pigments, carotenoids, anthocyanins, alkaloids. These are secondary metabolites are morphine, codeine, terpenoids, monoterpene, diterpene, essential oil, lemongrass oil, toxins, sorbin, ricin, lectins, concanavalin, drugs, vinblastine, curcumin, polymeric substances, rubber gums and cellulose. So these are the some secondary metabolites. Now secondary metabolites, what are these? Okay, when one analyzes plant, fungal, microbial cells, one would see thousands of compounds other than this called primary metabolites: alkaloids, flavonoids, rubber, essential oils, antibiotics, colored pigments, scents, gums, spices. These are called secondary metabolites. Now biomacromolecules, what are these? Those which have molecular weight less than 1000 Dalton and are usually referred to as micromolecules or simply biomolecules, while those which are found in acid insoluble fraction are called macromolecules or biomacromolecules. So these are the biomacromolecules more than 10,000 Daltons or are found in insoluble fraction, acid insoluble fraction. Now average composition of cells, so component the percentage of the total cellular mass taken by water, that is 70 to 90% proteins, 10 to 50% carbohydrates, 3% lipids, 2% nucleic acids, 5 to 7% ions, 1%. Now proteins, some proteins and their functions. So protein collagen function, intercellular ground substance, trypsin protein function, enzyme, trypsin, insulin protein, insulin function, hormone, antibody.